Hello everyone, this is Banabithi Bose. I'm a PhD student in Computational Sciences in Marquette University, Milwaukee, Wisconsin. Today I will talk about our recently developed computational tool named miRDriver that infers copy number derived microRNA gene interaction networks in cancer. As a background, from DNA via transcription, we get mRNA and microRNAs. MicroRNAs are short non-coding genes that do not translate into proteins. However, they can bind to mRNAs and influence its translation into proteins. As a downstream effect, biological processes can be affected. There were several studies, such as RegSys, Promise, ICPs, that were developed to find microRNA gene interactions. All these studies look for entire genomic locations for finding microRNAs, whereas in our study, we specifically look for DNA copy number aberrated regions. In copy number aberration or CNA region, a segment of DNA gets amplified or deleted. Studies have showed that these CNA regions can host cancer driver genes and cancer driver microRNAs. Hence, for this study, our hypothesis was copy number aberrated regions could host driver microRNAs that regulate downstream target genes that involved in key biological processes in cancer. With this hypothesis, in this study, we developed a computational tool called miRDriver that integrates copy number alteration data, DNA methylation, gene and microRNA expression datasets to compute copy number derived microRNA gene interactions in cancer. Our pipeline has 4 steps. In step 1, we found frequently aberrated CNA regions among cancer patients using a popular tool, GISTIC 2.0. We call these regions GISTIC regions. In each GISTIC region, we have 2 groups of patients, frequently aberrated and frequently non-aberrated. And we perform differential analysis using ADAR tool to infer differentially expressed genes among these 2 groups. We further considered these differentially expressed genes, that is DE genes, to be trans genes that reside outside a GISTIC region and cis genes that reside inside a GISTIC region. In our third step, for each trans gene, we collected all the possible cis genes and cis microRNAs from all GISTIC regions where that gene is trans. After this, in our final step, we selected potential microRNA regulators for each DE transgene using lasso regression. Since transgenes expression can be influenced by other regulators, we integrated copy number, gene expression, DNA methylation, transcription factors, along with microRNA expression datasets. Since the independent variables selected by lasso have shown to be inconsistent, especially in large cohort, we ran lasso 100 times for each transgene and selected the microRNAs that appear at least 70 times with non-zero regression coefficients, and in that way, we inferred our microRNA gene interaction network. We tested our pipeline on TCGA breast cancer and ovarian cancer datasets. Our tool inferred around 1800 microRNA gene interactions for breast cancer and around 1300 interactions for ovarian cancer. For the evaluation of our results, we compiled a list of known microRNA gene interactions from TargetScan, miTAR-based databases, which served as our ground truth data. We checked the enrichment of our inferred interactions with respect to our ground truth data and found that our interactions are enriched with significant hypergeometric p-values. Here's a short list of that. In the next, we compared our tool with 6 existing methods. Here I am showing the comparison results with RegSys, Hidden ICP and Promise. We started all this pipeline with same number of genes and microRNAs, and these are the number of selected microRNAs from each of these methods. Among them, we did the enrichment test only for those microRNAs that are present in our ground truth data and called them eligible microRNAs for enrichment. Then we took the overlap of miRDriver's predicted microRNAs with other methods. For RNA, there was no microRNAs that was significantly enriched in our ground truth data, whereas we have many enriched microRNAs for miRDriver's result in this overlap. And for all other cases, miRDriver outperformed them. We also checked the association of our selected microRNAs and genes with the survival of cancer patients. These figures are showing the adjusted Kaplan-Meier plots for two selected microRNAs within frequently aberrated group and non-aberrated group. These microRNAs are significant prognostic factors only within frequently aberrated group. We can see the significant log-rank test p-value only within aberrated group. We ran many other evaluations for our tool. For further information, please read our paper and visit our GitHub page for source code. I thank my advisor, Dr. Serdar Bozdag, for his guidance in this work. I also thank Bozdag lab members and our funding sources, and thank you all for your attention.